So, genome editing involves precise modification of an organism's DNA, and scientists can target specific sequences of DNA for removal or replacement. And this is offering a way to kind of correct genetic disorders. We can use it to enhance crops or even develop new treatments. So, one of the most well known technologies for genome editing is the CRISPR Cas9 system. So, the CRISPR portion stands for clustered, regularly interspaced short palindromic repeats. It was actually originally discovered in a bacteria. So, in a bacterial cell, the CRISPR serves as a viral defense mechanism and it allows the organism to remember and recognize previous viral invaders. The Cas9 portion represents an enzyme which acts like molecular scissors and is able to cut DNA at specific sites, allowing for the removal, addition, or alteration of genetic material. So Cas9 enzyme is very similar to the restriction enzymes that we previously talked about. So the CRISPR-Cas9 works by using a small piece of RNA, and this RNA is going to guide the Cas9 enzyme to the desired location in the DNA. Once the Cas9 enzyme reaches the target sequence, it's going to make a cut in the DNA, and then the cell's natural repair mechanism is going to either insert or delete the genetic material at this particular site. Transgenic organisms are those that have been genetically modified to include foreign DNA. So these organisms can be engineered for a variety of purposes, so things like producing medicine, enhancing food production, or even offering environmental benefits. So the foreign genes that are inserted into these organisms provide them with traits that would not naturally be found in species. So for example, transgenic plants can be modified to help resist pests or even to produce higher yields. So these GMOs, or genetically modified organisms, have really become an integral part of modern biotechnology. Genetically modified bacteria are also very important in biotechnology because we can insert specific genes in bacteria and then scientists can direct the bacteria to produce useful products. So one that we talked about previously was human insulin. That was a product of genetically engineered bacteria. Previously, insulin was derived from animals, but now we have these transgenic bacteria that can produce human insulin at really, really large scales, making it much cheaper and much more accessible for people who need it. Aside from medical uses, genetically modified bacteria can also be utilized in the environmental and industrial applications. So, for example, we can use genetically modified bacteria in bioremediation. So, for example, to help clean up oil spills, they can go in and they can break down harmful biocarbons that are present in the oil. Another example of the use of genetically modified bacteria is to help prevent frost damage in crops. So by inserting specific genes, these bacteria can actually produce proteins that protect the plants from freezing temperatures, helping reduce the risk of crop loss. Genetically modified plants have been developed really to help address several challenges in agriculture. So some crops have been modified to help resist insect pests, reducing the need for chemical pesticides. One good example of this is Bt corn. It contains a gene from the bacterium Bacillus thuringiensis that produces a protein toxic to certain insects. So it makes the corn resistant to pest damage. Other modifications have been made to improve the efficiency of farming practices. So we can have genetically modified crops like soybeans and cotton and corn that have been genetically engineered to tolerate herbicides, and that's going to help control weeds without damaging the crops. We also have crops like drought-resistant corn that have been designed to thrive in really water-scarce environments, making sure they can still produce high yields in regions that have limited irrigation. There have also been modifications aimed at really improving the quality of food. So for example, we have certain apple varieties that have been genetically engineered to help resist browning, increasing their shelf life and also reducing food waste. So overall, genetically modified plants have contributed to more sustainable farming practices. We've also been able to lower pesticide use and increase global food security. Genetically modified animals are another important area in biotechnology. So these animals are engineered to possess specific traits that are beneficial for food production, sometimes medical research, and sometimes for environmental purposes. So, for example, we have transgenic salmon. These have been modified to grow faster than wild salmon. So I'm going to show you the picture down here. So by incorporating a growth hormone gene from another fish species, these salmons can actually reach the market size much faster, making the fish farming more efficient and sustainable. In addition to food production, transgenic animals have also been created for medical applications. So we have transgenic pigs, for example, that have been engineered to help provide organs for human transplants. And these pigs are modified to make the organs more compatible with the human biology, and this potentially can alleviate the shortage of donor organs. We also have genetic modifications that are being explored to make animals more resistant to diseases like mad cow disease or foot and mouth disease, helping reduce the risk of outbreaks and livestock populations. So one of the more innovative uses of genetically modified animals is in a process that we have termed or coined farming, which involves creating transgenic animals that produce valuable pharmaceuticals in their milk. So one example is genetically modified goats. They can produce proteins like antibodies in their milk, and then we can harvest and purify these antibodies to be used in treatments for diseases like cancer. So this allows for the mass production of therapeutic proteins in a more cost-effective and sustainable manner. So farming is still in its early stages, but it does hold a lot of promises for future medicine by being able to offer cheaper and more accessible treatments for a variety of different conditions. So cloning is a technique often used in conjunction with genetic engineering to produce genetically identical animals. And this is going to involve the transfer of a nucleus of an adult cell into an egg cell that has had its nucleus removed. And the resulting organism is a genetic clone of the donor animal. So cloning can be used to replicate animals with desirable traits so that they can grow faster or so that they're resistant to diseases. So transgenic animals can also be cloned to ensure that offspring inherit specific genetic modifications. And this is particularly useful in agriculture, where animals with superior traits, such as increased size or disease resistance, they can be cloned to produce a genetically uniform population. So in biomedical research, cloning allows for the study of these specific genetic conditions and the development of the treatments that may otherwise be difficult to test.